How do we test for new bugs? Throughout the ongoing COVID-19 pandemic, a common refrain we've heard is, we need more testing, scale up testing. So how did we create the first COVID-19 test? And how can we make them better? Before we can test for something, we must first figure out what's making us sick. This wasn't always an easy thing to do. In fact, with tuberculosis, the oldest known infectious disease, it took many millennia before the causative agent was discovered to be a type of bacteria. With advances in science, we've been able to reduce this time from millennia to, in the case of COVID-19, just a few days. A key technology that's enabled this is genome sequencing. Human cells, as well as bacteria and viruses, have a genetic code that carries the instructions for how they behave, like a sort of cookbook of recipes. By reading out the genetic code, we can identify new organisms. Here I'm standing in front of one such instrument that reads the genetic code, called a sequencer. The flow cell I'm holding in my hand is where all the magic happens. An unknown organism is first inactivated, and the nucleic acids are then purified and broken down, or fragmented. This fragmentation is essential because most sequencers today aren't very good at reading. Sort of like if you were tasked to read an entire textbook in one sitting. You'd start off well enough, but without a break, you might begin to tire or get bored and start making mistakes. And so if we instead broke the textbook into smaller sections and had many of you read each section, we'd end up with far more accurate readouts. Same with the sequencer. This does mean that we have to put these sections back together. This is called genome assembly, and is a bit like piecing together a puzzle. If the newly assembled genome doesn't match with existing ones, we might just have discovered a new organism. And remember, with COVID-19, we made this discovery in just 5 days. Okay, so now we have the genetic code for SARS-CoV-2, the virus that causes COVID-19. With this information in hand, how can we design a diagnostic test? There are a few different ways of doing this, and one common way entails finding a small section of the genetic code that's unique to the pathogen we want to detect. Cookbooks and textbooks have some parts that are very similar, even identical, but there are other parts that make them unique. By comparing genetic codes from different organisms, we can find small regions that are unique to the pathogen we want to detect. Once we've picked such a region, we then use a test that makes many copies of this region, sort of like a copier machine. To make these copies, we borrow molecules that are present in bacterial cells, as well as our own, called polymerases. Polymerases in cells help with making copies of our genomes during cell division. By using this molecule in our tests, we can detect small amounts of genetic material. But since our tests aren't as sophisticated as the insides of cells, we rely on large instruments, such as this Thermocycler, which acts as the copier machine that detects tiny amounts of genetic sequences. It's the requirement of instruments such as this that make it so when you get a test done on yourself, you have to send the swabs and the samples away to a COVID testing facility. Could we bring this process closer to home? After all, microbes and cells in our body make copies of genomes all the time without needing large laboratories. Scientists, including myself, are working on such tests, where we take some more enzymes typically present in cells and create tests that only require a tube and a strip of paper. In many ways, these tests borrow from nature. Answers to fundamental questions of how life works and evolves can pave the way for technologies that help you test for COVID-19 at home.